Assalamu alaikum and welcome back to my YouTube channel Studyholic. This is gonna be the last lecture of cell organelles and this is also going to be lengthy. So I'll be adding the titles. You can skip to the organelles you want to understand. So let's begin with the lecture today. The first organelles are peroxisomes and glyoxysomes. They basically have two similarities with lysosomes. They are single membranous organelles and as well as they contain enzymes and originate from Golgi complex as that of the lysosomes. The both peroxisomes and glyoxysomes are collectively known as microbodies. Let's discuss first about peroxisomes. As I've discussed earlier, the peroxisomes and glyoxysomes contain enzymes. So the enzymes present in peroxisomes are some oxidative enzymes that are peroxidases, catalases, and glycolic acid oxidases. So how does the name peroxisome came? Uh, as the peroxisomes are abundant in liver cells, and over there in the liver cells, they are responsible for the formation and decomposition of hydrogen peroxide. Hydrogen peroxide. So as a result, the name came into being, that is peroxisomes. So peroxisomes are abundant in liver cells and over there responsible for the formation and decomposition of hydrogen peroxide. Now let's talk about the functions of peroxisomes. There are basically two main functions. First of all, it leads to detoxification of alcohol. So how does the peroxisomes detoxify alcohol? Now let's discuss about the procedure of the detoxification of alcohol. The first procedure, salt procedure, is basically taken place by the peroxisomes in the peroxisomes by the certain peroxisomes enzymes. The first step is the alcohol is basically converted into the hydrogen peroxide with the help of peroxidase enzyme. Over here this is alcohol and it is converted into the hydrogen peroxide within the peroxisome with the help of peroxidase enzyme. Then the hydrogen peroxide is basically a toxic molecule, so this immediately breaks down into water plus oxygen, and this reaction is carried out by the enzyme catalase. The second function of peroxisome is photorespiration. Photorespiration in plant cell. In humans, the peroxisomes are present in liver cells and over there they are involved in the detoxification of alcohol. And whereas if the peroxisomes are present in plant cell, they are responsible for the photorespiration. Not all of the procedure of photorespiration takes place in plant cell, but basically one step takes place in peroxisome in which the glycolate is converted into glycine with the help of enzyme called glycolic acid oxidase. This is glycolate. It is converted into glycine with the help of glycolic acid oxidase, GAO. Okay, now let's move on to the next organelle, that is glyoxysomes. Glyoxysomes are basically found in plants and they are found at the seedling stage in the oilseed plants. As I've discussed earlier, the glyoxysome and the peroxisome both have enzymes, and the glyoxysome has specific enzymes that are responsible for the plant lipid metabolism. That's over here, the lipid metabolism. Basically, why we say that the glyoxysome are present in seedling stage of the plants, because there are specific reactions that have to be taken place in the glyoxysome, and inside the glyoxysome there are certain enzymes responsible for it. As the germinating seedlings are basically, they convert the stored fatty acids to carbohydrate, and this procession from fatty acids to the carbohydrates takes place via different enzymes in the glyoxysomes. I have written over here The Fa stands for fatty acids and it is converted into carbohydrate in the seedling stage of the plant, and this is basically carried out by the enzymes in the glycosomes. And whole of the pathway from fatty acids to the carbohydrate is called as glyoxylate oxalate cycle. This is written as glyoxylate cycle. You can make the notes or you can also open the book to yourself and you can learn through this. That was all about peroxisomes and glyoxysomes. Let's move on to our next organelle, that is vacuoles. So vacuoles are basically large vesicles and they originate from endoplasmic reticulum, Golgi complex and plasma membrane. They are present in three locations, that are animal cells, freshwater protists and plant cells. So if we talk about animal cells, how in animal cells vacuoles are present? In animal cells, food vacuoles are formed by the process of phagocytosis. This is a cell, and through phagocytosis the food enters and the vacuole that results is known as food vacuole. Now if we talk about the freshwater protists, in freshwater protists there are many contractile vacuoles and their main function is to pump the excess water out of the cell, thereby maintaining a suitable concentration of ions and molecules inside the cell. Now we if we talk about the plant cells, in plant cells there are two types of vacuoles. Mean there is a young plant cell and a mature plant cell. And in young plant cells there are various small vacuoles, and they have reserves of important organic compounds. And as a result of these important compounds, the vacuoles in young plants help the plant protect the plant against certain herbivores. How the vacuoles help the plants against herbivores? Due to the compounds that are poisonous in nature or they are unpleasant to the animals. Now let's discuss about the mature plant cells. That how are the vacuoles in mature plant cells? Here is a diagram of mature plant cell and this is showing the vacuole. I'll be explaining it. As I talked that the young plant cell has various small vacuoles, and in a mature plant cell these all various small vacuoles combine and form a large vacuole that is also known as central vacuole, as you can appreciate over here. And the solution in this vacuole is known as cell sap, and this cell sap is reservoir of inorganic ions that are basically potassium and chloride. Now the central vacuole basically plays two roles. It maintains the turgor of the cell and also it acts as the storehouse of the cell. These are the two functions. Also there is a membrane between the cytoplasm, this is a cytoplasm, and the central vacuole, and that membrane that separates the central vacuole from the cytoplasm, sorry, this is not cytoplasm, that separates from the cytoplasm is known as. Now let's talk about the next organelle, that is mitochondria. Mitochondria is present in all eukaryotic cells and its number basically depends on the activity of cell. The cells, the metabolic activity, the cells that are more active have proportionally more mitochondria, they may be hundreds or thousands, and the cells that are less active has fewer mitochondria. Mitochondria basically capable of dividing themselves, that are they are self-replicating and they divide by fission. They basically divide by fission. Let's talk about the structure of mitochondria. Mitochondria are basically cylindrical or rod shaped and they are enclosed by double membrane, and that double membrane is in turn divided into the outer membrane and the inner membrane. Now let's discuss the two membranes in detail. Here is the diagram of mitochondria as it shows that it is cylindrical or rod shaped. There are two membranes. the outer and the inner membrane. This one is outer and this one is inner membrane. Let's discuss about the outer membrane first. I'm going to draw over here. This is the outer membrane. This is the outer membrane, a gross diagram. As you can see, the outer membrane is smooth in appearance, and it also has a somewhat like a sieve due to the presence of porins. These are the porins present in the mitochondria, and due to these porins, the mitochondria's outer membrane basically acts like a sieve. What are porins? Porins are basically special proteins, and they are responsible for the transport of the molecules across the membrane. Now let's talk about the inner membrane. Inner membrane basically looks like this, as I had shown into the diagram, and I am now drawing it and explaining it myself. Inner membrane, as its appearance shows, is folded inwards, while the outer membrane was smooth, and inner membrane is selectively permeable, whereas the outer membrane had porins and it acted like sieve. These folds are basically known as cristae. And these cristae are basically responsible for increasing the surface area of mitochondria. In the inner surface of membrane, the basically the inner surface of cristae, the mitochondria has granular particles, and these granular particles are known as ATP synthase. Here is the ATP synthase. This is shown. The inner membrane basically divides the mitochondria into two internal compartments. Further, it is divided into internal compartment. The first is the intermembrane space. This inner membrane space is present between the inner and the outer membrane. The second compartment is the mitochondrial matrix, and this matrix is basically enclosed by the inner membrane. This inner membrane, this encloses the matrix, this one, and this is known as mitochondrial matrix. Now, if we talk about the properties of mitochondrial matrix, it is basically a jelly-like material, and it contains small circular DNA and also all kinds of RNA, ribosome 70S, and enzymes. You can see the circular DNA, RNAs, and these components basically indicates that the mitochondria has its own genetic system. That means the proteins which are required by mitochondria are synthesized by their own metabolic machinery. They are not dependent on the others. Let's talk about the function of mitochondria. Basically, mitochondria is the site of cellular respiration. Cellular respiration basically is a pathway that uses oxygen to generate ATP. Oxygen is obtained and it is converted into the ATP, and hence the cellular respiration results in the ATP formation, which takes place in mitochondria. So the ATPs are formed in mitochondria, that are the energy, that are the basically reservoir of energy. And how the ATP is formed: not only from the oxygen is used and the energy is extracted from the sugars, fats, and other organic compounds that we intake. And this whole process takes place in the mitochondria by several enzymes in the inner membrane, the matrix, as the whole results in the formation of ATP. That was all about the mitochondria. Now let's move on to the next organelle. It is plastids. Plastids are basically present in plants and algal cells, and they are necessary for photosynthesis and food storage. Now I'll be classifying the plastids on the basis of the presence of pigments, that is proplastids, leucoplasts, chromoplasts, and chloroplasts. It is a diagram showing all of the classifications. Proplastids are basically young and immature plastids. They are self-replicating and they divide and redivide in the meristematic cells, and they are distributed to different cell types. The proplastids basically differentiate into the leucoplasts or the chloroplasts depending upon the the structures in which they are found, the intracellular factors, and on the exposure to the light. They are converted into the chloroplast or either leucoplast. So, what is the difference between chloroplast and leucoplast? The chloroplast are basically green plastids, and the leucoplast are basically colorless plastids. There is the third type as well, that is chromoplast. Now, let's discuss them in detail. The leucoplasts are basically found in the parenchyma cells of the roots, stems, and seeds. They act as storage organelles. As I said, they act as storage organelles. So, on the basis of the kind of the substance which they store, they are further classified into amyloplasts, elaioplasts, and proteinoplasts. These are the further classifications: amyloplasts, elaioplasts, and proteinoplasts. The amyloplast basically stores starch, elaioplast stores lipids, and proteinoplast, as it says protein, so it stores protein. I'm writing over here starch, lipids, and as the name says, protein. The chromoplast basically synthesize different pigments other than green. As the leucoplasts are colorless, chloroplasts are synthesize green pigments. So the chromoplasts synthesize different pigments, the color pigments. And as a result, they are found in colored parts of plant, that is flower petals and fruit wall, so that where they basically attract the insects and thus help in pollination. These are the locations, the petals and fruit walls. You can write it to yourself, or as I said, you can open the book and uh, retally from it. Now let's move on to the third one, that is chloroplast. Chloroplast are basically found in green parts of plants, and they are the basically site of photosynthesis. I'll be discussing the chloroplast in detail because it carries out the photosynthesis. Here is the detailed structure of chloroplast. I'll be explaining you every bit. Chloroplast is basically a discoid structure, as you can see over here. This is the structure, and it has three parts, that is envelope, stroma, and thylakoids. The chloroplast also has two membranes like mitochondria. The outer membrane is smooth like the mitochondria, and it also contains porins. So the outer membrane is freely permeable, and the inner membrane is semi permeable like the mitochondria, and it is rich in protein. And as the mitochondria, the chloroplast also has a space between the outer and the inner membrane, that is known as inner intermembrane space. As shown in the diagram as well, this is inner membrane, outer membrane. There is a space, this one, this is intermembrane space. Now let's talk about the component stroma. Here is the stroma. The stroma is basically colorless and it is proteinaceous substance, and like mitochondria, it also contains small circular DNA and all kinds of RNA, ribosomes, that is 70S, and various enzymes. In this stroma, there is also thylakoids, which is the main component of the chloroplast. The thylakoids are basically double membranous, and they are chlorophyll pigment bearing flattened sac like structures. These are the thylakoids, and there are two types of thylakoid, that is larger one and smaller one. These are the smaller one, and these are the larger ones. These are the connections are the larger one. Somewhere I'll be using layman terms, so kindly get the concept. And if you don't like my layman language, you can comment. I'll be discussing first about the smaller thylakoids. The smaller ones are disc-like as shown in the diagram, and they are piled over one another like the stack of coins. And each stack of the thylakoid, this is the stack, is known as granum. And if we say this stack as a whole, as a plural, it is known as grana. If we talk about each granum, so how many gross thylakoids are basically present? There are 25 to 50 thylakoids. 25 to 50 thylakoids, and about 40 to 60 grana are found in each chloroplast. Then we discuss about the larger thylakoids. Basically, the larger thylakoids connect the grana with each other. That are basically the connections. As I've shown, these are the connections. These are known as the large thylakoids. And they also have a second name that is known as intergrana. And these ones, the larger thylakoids, are colorless and they do not have pigments, whereas the smaller thylakoids have the chlorophyll pigment. We have discussed about the chloroplast structure in detail. Now let's discuss about the main function of the chloroplast. The main function is basically it is the site of photosynthesis, as I have told earlier. And photosynthesis basically has two phases, that is the light dependent and the light independent phase. These are the two phases. The first phase, that is light dependent. In this phase, the light is captured and it is transformed into the ATP. And this phase, the light phase, basically takes in the grana region of chloroplast. The second reaction, that is light independent, is also known as the dark reaction. In this phase, the carbon dioxide is basically reduced to make carbohydrates. And this reaction basically takes place in the stroma region of chloroplast. So the chloroplast is responsible for the photosynthesis. The light dependent the photosynthesis has two phases, light dependent and light independent. Light dependent takes place as grana and light independent basically takes place in the stroma. So that was all about the chloroplast. Now let's move on to our last organelle, that is centrioles. Today I'll be ending the organelles and will be moving on to the next topics of the book. Centrioles, basically, as you can see, they are non-membranous organelles and they are mainly found in animal cells. But apart from animal cells, they are also found in fungi like protists, such as slime molds and water molds. The centrioles, as you can see, they are rod-shaped structures and they basically occur in pairs. As over here also, a pair is shown, one centriole, the second centriole, and the pairs always occur at right angles. If the one is over here, that other would be at the right angle, that is this one, and they are found at the pole of the nucleus. So the basic unit of the centriole is microtubule, that you can see, and the microtubules basically occur in the triplets. These are the triplets, and in one centriole there are 9 triplets of microtubules. So what happens when the cell divides? Before the cell division, the each pair of centriole divides, this one and this one, and this results in the formation of two pairs. These are the two one, they will divide and lead into the two pairs, and these two pairs later move on to the opposite sides of the nucleus, and onto the opposite sides of the nucleus, the both pairs gives rise to the spindle fibers, the microtubules, during the cell division. I'm gonna draw over here this one. These are the two pairs of the centrioles formed during the cell division, and this will give rise to the spindle fibers during cell divisions, which we will discuss into the coming chapters. When I'll be discussing about the mitosis, meiosis, you'll better understand about it. And this whole apparatus which is formed during the cell division is known as mitotic apparatus, and this apparatus basically helps to arrangement of the chromosomes, the distribution of chromosomes between the daughter cells. So that was all about the today's lecture, a whole long lecture. You can please give me the feedback in the comment section. And now I'm moving on to the last session, the very important session as for me to know your concepts, that is brainstorming. Okay, so they are abundant in liver cells, peroxidases, peroxisomes, and oxidases. Think about the answer. Yeah, you would be knowing. It is peroxisomes. So listen carefully to this one. All of them are double membranous except the options are nucleus lysosomes, peroxisomes lysosomes, chloroplast lysosomes, nucleus or chloroplast. Choose wisely. The answer is peroxisome lysosomes. Moving on to the next one. All of them are single membranes except nucleus, chloroplast, nucleus, peroxisomes, lysosomes, peroxisomes, chloroplast, peroxisomes. It is gonna be easy one. Yep, nucleus, chloroplast. Moving on to our last question of this session, the organelle session. Which of the organelle always contains DNA? Centriole, Golgi complex, lysosome, mitochondria. This one is surely butter one. The answer is mitochondria. So, thank you for listening to me and keep supporting me. Thank you. Meet you in the next lecture.